Welcome to our third podcast on the digestive system. Today we're going to be talking about the intestines. So with the small intestines, we have three distinct regions that we will distinguish histologically between these three. Part of that will be identifying Brunner's glands in the duodenum and Peyer's patches in the ileum. Those are going to be the two landmarks we're really going to look for to help us define those. We're also going to compare and contrast the different cells that we're going to find in the different regions of both the small and large intestine, and we're going to compare and contrast the functions of each of those portions of our lower alimentary canal. So the small intestine is the longest single region of the alimentary canal, and small intestine may be upward of 23 feet in length. And the principal function of the small intestine is going to be for the absorption of the products of digestion. Now, the first portion of the small intestine, the duodenum, this is also going to be the completion of digestion. We're going to add in enzymes and materials from the pancreas, from the gallbladder, and this is going to help complete the chemical digestion, and then absorption will take place in the lower portions of our small intestine. So moving from superior to inferior along the path of our alimentary canal, our three regions are the duodenum, jejunum, and the ileum. In this illustration, we can see the three regions beginning here at the stomach. You can see the first small portion as the duodenum. The bulk of our small intestine will be the jejunum, or the middle portion, and then the last segment before we contact the colon is going to be the ileum. Now, in this illustration, there's a lot of information. I love pictures, and I especially love pictures that contain a lot of information. And so when we look at our intestine, and it's designed for largely absorption, we're going to see that we have three ways that we increase the surface area for absorption to happen. First, as we look in the small intestine, of course, we've got a long tube, so absorption is going to happen over a long period of time. But one thing that we also do on the inside, you can see these ridges, how they're folded up inside the intestine. These are called plicae circularis, or valves of Kerckering. And these are going to work almost like the fins on your washing machine to help move material down the intestine with peristalsis, but this also increases the surface area with the mucosal epithelial cells that we're going to have on the sides of these valves. So if we look at the valves of Kerckering, the plicae circularis, we can see here's one of the plicae circularis coming up. We can see on the sides that our mucosa is elevated. We can see these spike-like projections coming up. Each one of these spike-like projections is an intestinal villus. And so if we look more closely at an intestinal villus, we can see that we have a core of blood vessels and capillaries, and this green is going to be a lymphatic. That's a central lacteal. It's going to be important for transporting fats primarily. And on the surface of each one of these villi, you're going to have our columnar epithelial cells that have an apical modification of microvilli. We can see that here as our brush border. So between the valves of Kerckering, between the intestinal villi, and between the microvilli, when we add them up, we're not even adding in the length, but just these three modifications are going to increase the surface area of the intestine upward of 400 to 800 fold. So absorption of every bit of raw material we bring in our body is really going to happen in our small intestine. So look a little bit closely at these modifications. Of course, the plicae circularis, these are permanent structures. And we're only going to see that we have the first portion of the duodenum and the last portion of the ileum where you will not find any of the plicae circularis. So you can find these in each of the three regions, but specifically you're going to see them very well developed in the jejunum, or the middle portion. Now, just the plicae circularis alone, this is going to increase our surface area by a factor of 3, just counting for these specializations. Now, in a cross-section of intestinal tract, what we can see is we see our muscularis externa on the outside. We have our submucosa, but then notice how our mucosa is folded in. In some areas it's kind of small, but here we see an extensive fold coming in. Here we see another extensive fold coming in. These are cross sections through, each one of these being the plicae circularis. Now, the intestinal villi, which may be 1 millimeter tall, each one of these are also permanent structures. We already saw the capillary loops in the middle, and you may have some smooth muscle also in the middle of these intestinal villi to help extend and retract them as they're sort of kneading the material in the intestinal tract. The central lacteal, this is our lymphatic capillary. We saw that in the illustration as green. And as we look at them, they do differ in region. So we're going to have the greatest number of villi in the duodenum, and the tallest villi are going to be in the duodenum. Toward the end of this podcast, we're going to set up a table that describes many of these changes. Now, on these villi, this is where we're going to have our simple columnar epithelium that are going to have a brush border, the microvilli. We're going to describe them next. The intestinal villi will account for about a tenfold increase in the surface area in our intestinal tract. So here we have a scanning electron micrographic image of the intestinal surface, and here we can see our intestinal villi projecting up. And if we look at a freeze fracture through a longitudinal section of one of these villi, we can see our columnar epithelial cell, our microvilli, will be on this surface. And on the inside, this is where we have our capillary loops, our smooth muscle, our central lacteal. And most of these little round structures we see are going to be some of the lymphocytes that are present in this part of the lamina propria. At the light level, this is another longitudinal section. Again, our columnar epithelium. We're going to see increasing numbers of goblet cells as we get further down our alimentary canal. Here is going to be our lamina propria, a lot of plasma cells, lymphocytes. We've got some blood vessels here. We've got some smooth muscle here, and I'm not really seeing a good look at the central lacteal. In a cross section, we can see that central lacteal as a broad open area that's present in the middle of our intestinal villi. Now, lastly, we're not going to cover these a lot because we spent so much time on them with epithelium, but microvilli on the surface of our cells. And once we get to the intestines, we're going to call these surface absorptive cells, SAC. Remember, in the stomach, we had SLC for surface lining cell. Now, the microvilli, that's going to be the increase of 20. So if we take the 20, the 10, the 4, that increase, well, I think 3, that increase is going to come anywhere between 4 and 600 fold, depending on the extent of these modifications. And again, here we can see the plicae circularis, we see our intestinal villi, and we see the microvilli on the surface. Now we get into the histology. We were getting fairly good at looking at our gastric pits and gastric glands, and now we're going to see a very different appearance. Instead of looking in the stomach and seeing holes that are invaginations diving down into our mucosa, what we're going to find is we're going to have these stands of almost like a forest, with each tree being like an intestinal villi. And as we get down to the base of the trunk of the tree or the bottom of these villi, we're going to see that we have openings that extend further down into the mucosa where we have our intestinal glands, if you want to call them that. And these intestinal glands then we refer to as the crypts of Lieberkühn. This is just the bottom portion between our intestinal villi. Now, these crypts extend down through the lamina propria, and they're almost going to get down to the muscularis mucosa. That's the depth in which we extend these crypts. And we're going to have cells that are going to produce material, and this is called intestinal juice. And you may produce upward of 2 liters of intestinal juice per day. So if for some reason you have a parasitic infection or some kind of irritant in your intestinal tract, these intestinal juices are going to kick into high gear. You're not going to reabsorb that fluid, and so you end up with the condition called, that's right, diarrhea. Now, also within this material, we're going to have enteropeptidase. It's also called enterokinase. And this material is going to activate a pancreatic enzyme called trypsin. And the pancreatic enzymes and pancreatic material are going to be emptied into the intestinal tract, again, at the duodenum. And so trypsin is coming in. It is a generic protease, but we're also going to have amylase that's going to be coming in, and amylase is a carbohydrate degrading enzyme. So here's our illustration of our intestinal villi and intestinal crypts. Here we see the surface. This would be like the tops of the trees in a forest. And as we get down to the floor of the forest, the trunks of the trees, here we see the crypts are our intestinal glands extending downward toward the muscularis mucosa. In cross section, we can see our different cells. We have our SACs, surface absorptive cells, our goblet cells that are going to increase in number as we get closer to the colon. We're going to see some of our enteroendocrine cells. We've got our regenerative cells, and then we have some of these special cells that we'll talk about that are at the very base of our crypts of Lieberkühn. And these are really going to help us identify where we are if we're looking at the base of these glands and help us distinguish this base of a gland from the base of a gastric gland. So now as we look at the cells, in the mucosa, simple columnar epithelium. Most of these are going to be surface absorptive cell. We've got the brush border with microvilli. They're going to assist in the absorption and transport of material, including that we're transporting with triglycerides and fats. And so we talk about this being the terminal digestion phase and the absorption phase. And like we've seen everywhere else, we are going to have these tight junctional complexes to prevent this material from leaking between each one of these cells, getting into these deeper areas. But these epithelial cells are also going to have gap junctions. The purpose of the gap junctions, again, absorption being a global function that's happening over the entirety of our alimentary canal, this lower portion, this is going to help them function as a syncytium. Goblet cells, we've talked about those quite a bit. These are going to produce the mucinogen, that once it becomes hydrated, it is mucin. When it combines with the debris material, then it's called mucus. And we're going to see the smallest number of these in the duodenum, and as we move toward the ileum, and especially when we get to the colon, we're going to see these increase. Now, the goblet cells producing the mucus, this is for lubrication. We have other cells that are going to produce the mucus and the material that's going to contain the bicarb to neutralize the stomach acid. Our goblet cells are producing mucus just for lubrication. Now, we've already talked about the D and ES cells in the stomach. These are no different in the intestine. We just simply have fewer of them, but they are going to account for about 1% of the cells that are present in our intestinal tract. Now, the last type of cell that we're going to have is called an M cell or microfold. And these cells are going to be antigen-presenting cells. Again, they're part of this whole macrophage lineage. But when we see these M cells, they are going to be squamous-like cells, and you're going to find them situated above lymphoid nodules. Now, these lymphoid nodules we're going to find predominantly as we get toward the ileum. So these are going to be present in those regions. You're not really going to find these lymphoid nodules present in the duodenum or the jejunum. Now, as we get beneath the mucosa in the lamina propria, we have our typical loose connective tissue. We're going to have the core of the villi, but we are going to have a rich population of lymphoid cells, tons of lymphocytes, tons of plasma cells, and that's what we're going to see, a lot of these large numbers of very small dark staining nuclear cells. We will have our crypts of Lieberkühn extending down, and so these are our intestinal glands. So we've got these simple tubular glands that have the goblet cells, the surface absorptive cells in the upper portion, regenerative cells, D and ES. But these Paneth cells, these are new to our glands that we see down in the crypts of Lieberkühn. Now, these Paneth cells are producing lysozyme, and they are produced and stored in these fairly large eosinophilic, which are red in color, granules. And these are going to be produced and secreted into the crypts. And they may be around for 20 days or so. But as you can see in this micrograph, we can see our other cells present in the base of our crypt of Lieberkühn. But then you're going to have these humongous red granular cells that are present at the bottom. These are going to be so hard to miss. If we look in a different micrograph, again, this is a cross section through a crypt of Lieberkühn, but you can see many of these columnar cells, and then all of a sudden you see these red granules just absolutely filled with these red granules. This is possibly one that doesn't quite have as many granules, but Paneth cells are going to be pretty difficult to miss. Now, the muscularis mucosa, this is really going to be the extent to where we will see our crypts of Lieberkühn extend. And so we're going to have our smooth muscle layers. We are going to extend some of these up into the villus. So the smooth muscle from the muscularis mucosa will extend upwards. And so again, these are going to help with the contractions, shortening the villus, extending it, almost like kneading bread. If you've ever made bread and just start pushing your fingers in and out of that bread, that's what's happening with these contractions of these villi. Our submucosa, for most of what we're looking at, it's going to be fairly unremarkable. Dense, irregular, fibroelastic connective tissue. We're used to seeing this. And in the jejunum, that's all you're going to see. However, in the duodenum, and so we sometimes refer to them as duodenal glands, we are going to have Brunner's glands. Now, these glands are going to take up much of the submucosa. They're branched tubuloalveolar glands. They're going to be very rich with this mucus acinus. And so this mucus material is going to empty into the base of the crypts. And so this mucus is going to be alkaline in nature, and it's going to be the secretion from these glands that's going to neutralize the hydrochloric acid coming from the stomach. And so that's where we neutralize the acid, not the goblet cells that are producing that mucus. But we're also going to see that cells in Brunner's glands also produce a substance that's called urogastrone. And urogastrone is going to inhibit the production of hydrochloric acid. So as material is emptying into the duodenum, we're pretty much completing the chemical digestion in the stomach. So here's our negative feedback. To shut down hydrochloric acid production in the stomach, shut down chemical digestion as we're processing and neutralizing the acid to move the material further down the alimentary canal. So here we have a section of intestine. We can recognize this because we've got our intestinal villi. You see this quite a bit of space between each one of these. And in most of these, we're going to see an open space in the very middle. That's going to be our central lacteal. Then at the base, where you see that we have a coming together of this material, here, this is going to be our crypts of Lieberkühn in this area. Here we have our muscularis mucosa, and then all of a sudden you see this expanse of what looks like completely mucus acini in these tubuloalveolar glands. And so these are Brunner's glands, and when you see this in the submucosa with intestinal epithelium, you know you are in the duodenum. Now, as I've mentioned, we're simply going to have our submucosal connective tissue in jejunum. So if you don't see mucus glands in the submucosa, and you don't see Peyer's patches in the submucosa, then you know you're looking at jejunum. Now, Peyer's patches are lymph nodules that are going to be present in the ileum. Now, these are, for all intents and purposes, in the submucosa, but as you can see, they may extend up into the mucosal lamina propria as well. But these are permanent structures. So if you see intestinal villi and you see lymphoid nodules in the submucosa, then you're in ileum. But let's say, for instance, this is what the entirety of the intestinal tract would look like on this side. You don't see any Brunner's glands. You see no lymphoid nodules. Then you would know you are in the jejunum section of the small intestine. And that's how we distinguish those three regions. It's pretty easy. Muscularis externa, we have our same layers. All of the small intestine is going to be serosa, except for a small part of the duodenum, which is going to be adventitia. The first part is still going to be serosa from the mesentery of the stomach. We're going to have this second and third portion of the duodenum before we get to the jejunum, which is going to be uncovered. It's not going to be within the peritoneum, but then the remainder of our small intestine will, in fact, be covered and be a serosa. So here we have a chart that I've put together. We have our three regions, the duodenum, jejunum, ileum. When we look at the villi, these are going to be the tallest. They're going to be the most broad and the most numerous. As we move downward, notice we shorten the villi. They become thinner, almost finger-like, and they become fewer in number, with the fewest being in the ileum. Now, goblet cells, on the other hand, are going in the other direction. We have a few of them in the duodenum and most in the ileum. With lymphocytes, you may see some lymphocytes in the duodenum, but this is really what we're looking for. Peyer's patches in the ileum, and those are clearly recognizable. Our glands that are present, mucus glands, that's going to be totally in the duodenum. We're not going to see that. And then the connective tissue, we're really not going to be looking for that. We're looking for Brunner's glands, we're looking for Peyer's patches, and we're looking for intestinal villi to help us distinguish the regions of our small intestine. Now, the small intestine brings us to another sphincter, and this is going to regulate the passage of the material from the small to the large intestine. So this is called the ileocecal or the ileocolic valve. And once we pass this sphincter, now we're in the large intestine, which is going to be approximately 6 feet long. So we usually say the intestinal tract is about 30 feet in the average human. Now, in the large intestine, we have regions, just like we saw in the small intestine. The first portion is going to be the cecum. Almost think of this like a T intersection. If you turn to the left, you're going to end in a dead-end street. That's the cecum, and we're going to have the appendix coming off of the cecum. However, if you turn right, so you're heading in a superior direction, you're going to be moving into the ascending part of our colon, and we'll look at a picture of this in just a bit. But the ascending part typically goes up the right lower quadrant of your body. Then it goes across your body as the transverse portion, descends down to the left lower quadrant. Then you have this S-shaped bend, the sigmoid portion, that brings your colon to the midline where it connects with the rectum, and then the opening to the outside of the body, which is the anus. Again, in our illustration, we can see our colon. This is ascending, transverse, descending, sigmoid. Here's the rectum with the anus at the very end. You can see the ileocecal valves about in this location, and this portion, the dead end portion, that's the cecum, and we can see the appendix sticking off, the vermiform appendix, and vermiform means worm-like. So once again, as we look in the colon, we can clearly see our crypt. As we look in the colon, we can clearly see our crypts of Lieberkühn, but what we're missing are the intestinal villi. Those are absent once we get to the colon. And when you look at the cells of the crypts of Lieberkühn, we do have the absorptive cells absorbing mostly water at this point. Tons and tons of goblet cells. Those will be very numerous. Regenerative cells, enteroendocrine cells, those are going to be present here. We will not see Paneth cells in our colon. So, abundant crypts, no villi, no Paneth cells, and we're watching the goblet cells increase in number from the cecum to the sigmoid colon. There we have our surface absorptive DNES and our regenerative cells, which are continuing to turn over very quickly, 6 to 7 days versus 5 to 7 days in our stomach. We don't have as harsh conditions as far as acid and enzymes, but as we move through the colon, we're desiccating the waste, and so we have frictional stresses on these cells to turn them over more and more quickly. So here we have in the colon, we have our crypts of Lieberkühn. Notice we don't see any villi projecting off of the surface, and most of what we see in these white areas, those are going to be remnants of goblet cells. They extend down to the muscularis mucosa. There we see our lamina propria. And in looking at a higher mag, we can see again goblet cells, absorptive cells, regenerative cells. Here's an enteroendocrine cell. And you may look at that and think, oh, well, there's a Paneth cell. Well, look where it's located. It's not within the gland. It's in the lamina propria. That's actually an eosinophil. Now, as we look at other cells, here is one of our classic ones. We learned this very early in our connective tissue. But notice how we have the dot in the very center, the material on either side. That is our clock-faced nucleus. That's typical of a plasma cell. And you can see that plasma cell, there's a plasma cell. Plasma cells are going to be all over the place through the lamina propria here in the colon. Now, our colon has a very interesting design. When we look at the muscularis externa layer, that smooth muscle layer, in the colon, we're not going to completely form the outer longitudinal layer. Instead, it's going to be arranged in 3 longitudinal strips of skeletal muscle that are called the taenia coli. Now, these ribbons, they're going to keep the colon in somewhat of a constant state of folding or contraction. And as we do, we're going to have these pleats or folds, they're referred to as sacculations of the colon. And notice how they always draw it sort of bumpy. And those sacculations are called the haustra coli. So in this illustration, we can see our taenia coli, 1, 2, 3 ribbons of smooth muscle, and you can see this folded nature, and these are the sacculations or the haustra coli that basically confirms the appearance that we're used to seeing with the colon. Serosa, this is going to be the outermost layer, and it surrounds the entirety of our large intestine. So we have a Lomax shot, again, looking at the colon. Mostly what we see, all this white, that's going to be our crypts of Lieberkühn, and we have that white appearance because of all the goblet cells present. Muscularis mucosa is in here somewhere, we don't really see it. Lamina propria. Submucosa, for all intents and purposes, is going to be fairly thin in most places, but then we get to our muscularis externa, and these knobs that we see are our cross sections of our taenia coli. So those knobs that we see out here, that's going to be the indicative histological feature that we look for to help us identify even this low magnification view and be fairly confident we're looking at colon. Now, as I've mentioned before, the function of the intestine is absorption, but the large intestine is principally going to absorb the water and all of the fluids and juices that we produce. It's also going to be a location where we compact the feces and we prepare the feces for elimination, approximately 100 mils of feces a day. Now, the last thing to mention, only because it comes up when we have problems, is our vermiform appendix. This is going to be a diverticulum that's off the cecum. It may be 5 to 6 centimeters long. Usually, you're not going to see any villi because it resembles what we're doing in the colon. We're not going to see placa circularis, and if you see crypts, they're going to be fairly shallow. But what you will see very often in many sections is debris in somewhat of a collapsed lumen, but you're also going to see lymphoid nodules. And so if you sort of look at the lymphoid nodules and let that be somewhat of a guide to you as far as the scale with which you're looking at, you can see that we've got 1, 2, maybe 3 or 4 lymphoid nodules, and that's pretty much the diameter of the lumen that we see here. In Peyer's patches, we had many, many more than that extending across the lumen. So we know that this is a fairly small structure as far as a luminal diameter. You can also see the white spots, which are going to represent some of the crypts with their goblet cells. And so this very small luminal structure is going to point out to you, especially with these lymphoid nodules, that you're looking at the vermiform appendix. This most certainly is not Peyer's patches present in the small intestine. We don't see intestinal villi, and it's not nearly big enough for that. Now, as we get to the end, we get to the rectum. The anus is the opening, and what we're going to see is another transition similar to what we saw with the lip. So in this case, we have our colon material coming from the right. We have our typical colon type epithelium, the mucosa. Here we have our submucosa, lamina propria, muscularis. And as we get out here to the end, we can see how we transition that mucosa from the rectum at the anus, and this is going to be the surface of our skin that's on our buttocks, and we can see hair follicles and everything we recognize as thin skin. So we know that this is going to be the place where we're going to have this expanded muscle, and this expanded muscle is going to be our anal sphincter that controls the release of our feces.